All right, so this time we're talking about translocation. So not transpiration, translocation, which is the movement of assimilates throughout the plant. So it could be things like, we'll be focusing really only just on sucrose, but it could be other things like amino acids as well. So focus so far has been, we've been focused on the xylem and how water moves up the xylem from roots to the leaf. Now we're focused on the phloem, and phloem, although this only shows it going down, the fluids, and it's really just a sap, it can move down the phloem and it can move up. So I'm going to go into detail about the mechanisms that cause this movement to take place, because there is no pumping heart or anything like that. Mechanisms are in place to force the fluids to move in different directions. So phloem, they're similar to xylem, no organelles, no nucleus, no mitochondria. So these individual cells that make up the phloem vessel, we call those sieve tube elements. They can't really do much for themselves, so they need help from a neighbouring cell called the companion cell. So anything that requires energy, like active transport, it will require the help of the companion cell. So to go into detail about how movement takes place, we're going to talk about the idea of what a source and what a sink is. We'll go into detail, and we need to go into quite good detail about how the companion cell plays its part. Sieve tube elements, the process of active loading, active transport, what co-transport is. We're going to focus on sucrose. We're going to talk again about water potential and how this is being manipulated to cause osmosis, and how hydrostatic pressure is being created, and eventually how this causes mass flow to take place. All right, so have a look at this image here. You can see the two main vessels are the xylem and the phloem. Xylem is carrying water and mineral ions. They're moving up. The phloem, you can see there's water going in, but it's going both ways, up and down. And not all parts of the plant can produce sucrose or glucose, but every single part of the plant needs to be able to respire. So if you think about the roots, they've got an important job. If the root hair cells, they need to be able to actively transport mineral ions in. Well, where are they going to get the ATP to do that job? Those root hair cells will need to be able to respire. And to respire, they're going to need glucose. So the glucose that's been made in the leaf needs to go down to the root so that it can produce its own ATP. So how is that glucose getting down there? So first of all, it's not been transported as glucose, it's been transported as the disaccharide sucrose. All right. So here is the sucrose in the palisade cell. This is where it was created by the chloroplasts. Because this is where the sucrose started, we call this the source, and its destination is the sink. So when we talk about translocation, we're talking about the movement of assimilates from source to sink. The sucrose needs to be able to leave the palisade cell, get into the companion cell, and then finally move into the sieve tube element where it can be carried away to the sink. Right, we need to understand how this is done. So we've got quite a few different players here to discuss. We've got the palisade cell, the companion cell, sieve tube element, and the xylem. Right, so let's start off with the palisade cell. As it's undergoing photosynthesis, it's producing sucrose, just the letter S for sucrose. And as it produces more and more and more, there's a concentration gradient. There's a high concentration, but it's unable to diffuse into the companion cell because the cell membrane is impermeable to sucrose. The only way sucrose can enter the companion cell is through co-transport. And co-transport it needs to bind on basically with a hydrogen ion. And those hydrogen ions are going to come from the companion cell. So the companion cell is actively transporting hydrogen ions out into the source. This requires ATP. So this is active transport. As soon as enough hydrogen ions are pumped out, there's a high concentration of hydrogen ions out here. They are going to then bind with sucrose. And once this happens, the protein channels will allow sucrose to enter, but only when it's binding with the hydrogen ion. So it's the protein's tertiary structure has been slightly altered with the presence of the hydrogen ion, and now it can enter. And this is just diffusion now. There's no active transport needed here, but it's not simple diffusion. This is a facilitated diffusion or co-transport. As soon as the hydrogen ion and the sucrose enter, they separate from each other. So they're separate again. And this hydrogen ion here, this is ready now to be pumped back out again, so they can maintain a concentration gradient. If all the hydrogen ions eventually diffuse in, you'd get equilibrium. So energy is needed to maintain this concentration gradient. Once the sucrose keeps entering with the hydrogen ions, concentration gradient is going to build up, and it's unable to diffuse back out where it came from because, remember, this cell membrane is impermeable to sucrose. The only reason why it entered was because it went via a protein channel, which only allows it through if it's in the presence of hydrogen ions. However, this side of the companion cell next to the sieve tube element is permeable to sucrose. The sucrose can just diffuse in here now. So our sucrose is building up. Now this sieve tube element, or this phloem vessel, contains lots of water as well. And as more and more sucrose enters, well, sucrose is a solute, so it's going to lower the water potential inside this sieve tube element. And if it lowers it enough, we're going to get a water potential difference. You're going to have a low water potential in the sieve tube elements and a higher water potential in the xylem. And if that occurs, well, osmosis is going to happen. So water is going to begin to leave the xylem through the bordered pits and enter the sieve tube element. Osmosis is the movement of water from a high water potential to a low water potential across a partially permeable membrane. As water keeps entering, more and more water will fill up into this sieve tube element, and this is going to cause a high hydrostatic pressure to occur. And if the pressure continues, so we've got our high hydrostatic pressure, that pressure, as it builds up, is going to be pushing up against the walls of the sieve tube element, and eventually the water is going to be forced away from this area. So let's focus on, because we looked at this image before here, we can see that it's going down. We can assume that the water has the sieve tube elements down here have a low hydrostatic pressure. So the water is going to move from a high to a low hydrostatic pressure. So it starts to move down to the roots. And as it moves down the roots, we call this mass flow. It's going to bring with it all that sucrose, which is needed by the roots. Right. So why is there a low hydrostatic pressure down here? Well, I'll just show you this picture here. What's happening? You can see the sucrose is leaving the the sieve tube element, or the phloem vessel, and it's entering another companion cell where it's then been transported into could be the root hair cell, for example, which can use it for respiration. So if the sucrose is being unloaded, or it's been moved out of this sieve tube elements, it's going to increase the water potential down here. Because there's less solute, the water potential is going to go up. And if the water potential goes up and up and up and up, there'll come a point where the phloem has, as to the letters, has a high water potential compared to the xylem, low water potential. So water begins to move from a high water potential to a low water potential. So this difference in water potential is being created by the loading, or the active loading, of sucrose at the source, and suddenly the water potential has been increased because the sucrose has been unloaded, and this is going to keep going on and on. So it doesn't necessarily have to be that the leaf is the source. It all comes down to, where is the sucrose? So in the summer, when the plant, let's focus on a potato plant, when that potato plant is photosynthesizing in those warm days, lots of sucrose is being transported from the leaves down to the potatoes in the roots. However, in the winter, when there's not enough sunlight for the plant to photosynthesize and the leaves are gone, the potatoes, the roots, become the source, and it switches around. So these companion cells can do what they were doing down here, what they were doing up here, they're now doing down at the roots. So this companion cell it's able to take sucrose out of the sieve tube elements, or it can load it in, depending on what the scenario is. If there's a need for sucrose at the top of the plants, then it's going to switch around. Also, even at the leaves in the summer, it could still be moving up, because you've got to think about different times of the year. If, for example, an apple tree is producing lots of apples or flowers, lots of sugar is needed for that process to take place as well, like the nectar and all the sugar in the apples as well.